In this video, we'll address how the brain transforms sensory impressions into the world we experience through concurrently analyzing distinct categories of information. The brain's ability to simultaneously analyze distinct categories of incoming sensory info is called parallel processing. When discussing the information processing approach, we considered a linear model moving from inputs to invisible cognitive processes to outputs. This serial processing portrays cognition as a series of linear sequential steps, whereby the earlier step is complete and then followed by the next step. Parallel processing, in contrast, portrays cognition as a series of simultaneous processes, each working on discrete kinds of information. As a footnote, another model called parallel distributed processing, or what is sometimes referred to as connectionism, also presents cognition as a series of simultaneous processes. However, in that model, the processes are not independent but provide constant feedback to each other. Let's see how parallel processing works for visual perception. We'll begin with a cluster of cells called the Lateral Geniculate Nucleus, or LGN, which is located in the thalamus. The LGN receives connections from the optic tract. And when we get an even closer look, we can see that the LGN is made of a series of striations categorized as belonging to the Parvocellular layer or the Magnocellular layer, each consisting of P cells and M cells respectively. We can begin to see the parallel processing model at work here, as each cell has unique responsibility for processing specific visual information. P cells specialize in processing color and discriminating fine detail. In contrast, M cells process information related to depth, motion, and the localization of objects, including their orientation and position in space. M cells are theorized to be underdeveloped in some reading disorders, what is called the Magnocellular theory, since they're involved in stabilizing a visual image to compensate for minute eye movements, helping to focus on a single object and blur the less relevant surrounding environment. Dysfunction in these cells can lead to some deficits observed in processing individual letters, for example. But despite some research supporting this theory, it does remain a controversial explanation for reading disorders. Even at an early stage of vision, parallel processing is taking place. This continues when that information going to the LGN then moves on to the occipital cortex at the back of the brain. The occipital cortex is divided into several distinct functional areas. And we begin with area V1, which is in the primary visual cortex and is perhaps the most studied region of the brain's visual areas. V1 identifies what is important among all the visual inputs and uses this information to guide visual attention. The neurons in this region are specialized in detecting contrast in light and dark that allow for edge detection. Edge detection is important for identifying individual objects in our visual field. Moving on to the secondary visual cortex, area V2 receives direct connections from V1. V2 processes relatively more complex aspects of the visual field, helping to distinguish the figure from the background. It also plays a role in guiding attention to important information and helping form memories of visual objects and recognizing them. Next are visual areas V3 and V4, and here we begin to see another set of parallel processes take place. Area V3, especially the top or dorsal portion, appears to be involved in general motion processing, whereas area V4 continues the work of V1 and V2 by processing an increasingly complex set of features related to objects and is even more strongly involved in directing visual attention. V3 has direct connections to area V5, or what is more commonly called MT, which also receives direct connections from V1 and V2. Area MT processes the more complex dimensions of motion, such as direction and speed. One particularly devastating condition thought to result from damage to this area is called Akinetopsia, or motion blindness. An individual with such a condition is described as experiencing the world like a series of still images. leading to difficulties in estimating the distance of moving vehicles or trouble predicting when a cup will be filled upon pouring water into it. One particular illusion, called the phi phenomenon, demonstrates how our brains process motion, using this information to identify individual objects, even when in fact those objects are separate, as can be seen here. However, individuals with Akinetopsia are unlikely to undergo this illusion, experiencing the traveling light as a series of separate objects rather than as one object traveling in a circle. From here, we can see the divergence of two major visual pathways, each responsible for processing a distinct aspect of our visual fields. Together, these make up what are called the two streams model, which proposes two qualitatively different cognitive systems for processing sensory inputs. First, there is the what, or ventral pathway. This pathway carries visual information from the visual cortex to the temporal lobe. The what pathway is responsible for recognizing the objects we see, connecting the processed visual features of objects to our stored mental representations. Without this ability, the world would always seem unfamiliar, as we would not be able to recognize everyday objects as we ordinarily do. Keep in mind that this is not a problem with memory, but of connecting visual inputs to memory. So someone shown a key may not be able to identify it, but they know what a key is, and upon putting the key in their hand, they can recognize it. This condition of not being able to recognize something that you see is called agnosia, and we'll return to it when we discuss the topic of recognition. The second major pathway is the where, or dorsal pathway. Here, information travels from the occipital lobe to the parietal lobe. While it's called the where pathway, it could also be called the how pathway because it's responsible for recognizing where objects are in space as well as guiding our motor movements in how to interact with that object. Damage to this pathway can lead to several conditions. One, called hemispatial neglect, involves problems with spatial attention. Another condition that can occur is called apraxia. Someone with an apraxia has difficulties executing a sequence of voluntary motor movements that were previously learned. It may involve the loss of the ability to brush one's teeth, make coffee, or giving a thumbs up. This is not a problem with motor functioning specifically, as the ability to move remains intact. It's also not involving a problem with understanding what to do, as the person may be able to verbally report the necessary steps involved in some motor task. Rather, it's about coordinating the stored mental representation of a motor sequence with the motor region responsible for executing that task. Spatial information is often an essential component to this process, knowing where and how to move in space. We've so far considered how categories of sensory input are simultaneously processed in different parts of the brain. This raises a major problem, and it's one so fundamental that it concerns nothing less than the nature of consciousness itself. My experience of the world does not seem like a series of discrete sensory inputs. When I see a car, I don't see its shape, color, and motion as separate elements of my experience, but rather as a single object. But given that the brain processes these categories in different parts of the brain, what's the mechanism that ultimately brings the pieces together to form this unified experience I'm having right now? Also, in a related issue, when I'm having an experience of two objects, let's say a blue triangle and a red circle, what allows me to perceive these shapes as having these colors and not instead perceiving the triangle as red and the circle as blue? This is known as the binding problem. Our everyday experiences consist of unified objects. We ordinarily don't experience color absent of structure and motion without an object. There are some exceptions, however, and these exceptions might shed some light on the problem. One exception are known as conjunction errors. When perceiving two separate objects, we ordinarily don't mix up their colors, but under specific experimental conditions, this has been demonstrated to happen. Such a study would have the participants focus on an object at the center of the screen while engaging in some task, perhaps focusing on how many times that object flashes or to identify when its color changes. At the same time, participants were shown simple colored objects on the screen that were outside the main area of focus, but they were still within the peripheral vision of the participants. They were consciously aware that these objects were present and they could accurately identify their features. However, the interesting thing was that the participants were far more likely to confuse what color belonged to which object. This research finding, among others, supports an explanation for the binding problem called feature integration theory. According to this theory, sensory inputs are processed in a series of stages. First, the constituent components of the object are analyzed, what is known as the pre-attentive stage. Next, the individual components just processed are combined through focused attention. This finally leads to the conscious perception of a unified object. In the situation where a conjunction error has occurred, the lack of focused attention on objects in the periphery leads to their individual components being processed but without any consistent integration. So one answer to the binding problem is focused attention. But how does attention do this neurologically? Neural synchronization theory provides another solution to the binding problem, one that may complement feature integration theory. According to this explanation, sensory inputs, while processed in distinct brain regions, lead to neurons in those regions to fire together at a similar rate. This rhythmic synchrony is then registered by the brain as a unified experience. Focus attention in a specified location of the visual field may be what allows these distinct areas to fire at similar rates. As is the case with most explanations provided in this video series, the binding problem is far more complex and nuanced than presented here. And the solutions presented are not the only ones, and they're not without dissent from cognitive scientists, in particular Daniel Dennett, who makes the radical claim that conscious experience is in fact not unified, but rather a grand illusion of the mind. It's constructed out of its tendencies to cover over the gaps of experience. Thank you for watching this video. If you've enjoyed it, feel free to subscribe so you can know when the next video is uploaded. I'll see you next time.